Thanks so much, Randy. And I want to thank the planning committee for putting this wonderful opportunity and this conference together, and for inviting me to come. It's just really nice. As Randy mentioned, I did my college and graduate studies in Texas. I am now up at the Michael J. Fox Foundation in New York. So it was nice to come south again, be back. I mean, yes, it's a little humid, but it is great to be back in the south. Okay. So yes, this is. I acknowledge this is a slide full and a mouthful and a somewhat intimidating title, but the point that I want to make and what I will talk to you about today is what we all know. Everyone in this room knows that we need improved therapies, and we need to learn more about what it will take to get a cure for Parkinson's disease. Many of you take the levodopa or the Sinemet, the medicine that replaces some of that dopamine that Dr. Vaillancourt mentioned. You need dopamine for movement. You need dopamine for many of these functions that are important. And when those cells that are affected in Parkinson's disease die, they don't produce as much dopamine. And so currently the therapy is replacing something that's missing naturally. And while that treats the symptom of Parkinson's disease, that doesn't treat what caused the cells to die in the first place. We call something that would treat the cells that die a disease-modifying therapy. And that's what we're really working towards, because the current therapy of just replacing the dopamine has not changed for decades. It's still the same procedure. Little things change here and there, and we'll learn more about that later. There are improvements on how to keep the levels of dopamine more steady to prevent side effects, and there's a lot of exciting efforts in that area. But what I want to talk about today is how we are studying, and how the Michael J. Fox Foundation and how pharmaceutical companies are really working hard to try to find a therapy that actually changes the disease in a positive way. That's a very hard thing to do, but I'll show you and tell you how we're doing that. Specifically, we're looking at something called alpha-synuclein. Alpha-synuclein is just a protein that's found in the dopamine cells in the brain. It's also found in other places. But I will explain to you why Parkinson's disease is implicated, how alpha-synuclein is important in Parkinson's disease, and I'll explain to you why we are targeting it for improved drug therapy in a way that we think will actually change the disease for the better. So I'll talk to you about our strategy for targeting alpha-synuclein. First, I want to show you how this alpha-synuclein protein falls into the overall research priorities and the strategies that we have at the Michael J. Fox Foundation. I mentioned that we're trying to find something that is disease modifying, that changes the disease. And alpha-synuclein is part of what we see as the main targets for disease modifying therapy within the Michael J. Fox Foundation, along with some other targets. We also have a lot of research around symptomatic areas. So of course this is looking at non-motor symptoms, also at motor symptoms. And then we also work on field-wide challenges. We work on improving clinical trial recruitment. We work on connecting people, building bridges, and also something called biomarkers. Dr. Vaillancourt was talking about how we need to be able to measure outcomes. We need to be able to look at what's changing in Parkinson's disease. And there's a big need for better biomarkers. So biomarkers just, are an indication of something that's changing in the body. For example, if you have high blood pressure, that's a biomarker for heart disease or stroke. You might go to the doctor because you have a cold, but they check your blood pressure anyway, and if they see routinely that you have high blood pressure, then they might put you on medication to bring your blood pressure down. Well, if you go to the doctor maybe 3 or 4 months later after taking the blood pressure medicine, and your blood pressure is down, that's how that biomarker was used first to identify you as at risk, and then secondly to identify that the drug was working. So we need biomarkers for anything that we're looking at that could be potentially changing the disease. We need to be able to tell if that marker, that biomarker, is also changing. We also need to find biomarkers that will help us diagnose earlier, and that will help us to evaluate whether or not a test drug is actually doing what it needs to do. So that's why I'm going to talk to you about specifically modifying therapies for alpha-synuclein and biomarkers for alpha-synuclein, because if we're going to target it as a therapy, we need to be able to measure it. So at the Michael J. Fox Foundation, we prioritize all of these research areas and use a team approach, a combination of scientists business strategists, and the outside expert scientific research community to move forward with these goals. So now I'm going to dig in and give you the reason why we're targeting alpha-synuclein. It's not just because it has this funny name. There's a really strong indication from genetics that alpha-synuclein is important in Parkinson's disease. First of all, it's shown that multiplication of alpha-synuclein can lead to Parkinson's disease in a genetic inherited way. What's really interesting about this is that the different ways that alpha-synuclein mutations can occur lead to the disease. Even though the starting mutation is different, the result is the same. So some people have a mutation where they just make too much normal alpha-synuclein. Too much normal alpha-synuclein can lead to Parkinson's disease. But you can also have mutations in alpha-synuclein that make it have a change in its function. So abnormal alpha-synuclein also leads to Parkinson's disease. So that's a strong indication that alpha-synuclein is very important in the disease, because too much of the normal is bad, but also just having abnormal alpha-synuclein is bad. Now that's what genetic studies tell us, but that doesn't really tell us biologically or pathologically what the alpha-synuclein is doing. But we do have some more information on that. It turns out that alpha-synuclein is a major component of Lewy bodies. Lewy bodies are these spheres that we see if we look at the microscope and we look at a brain under the microscope. A patient with Parkinson's disease will have a lot of these dark spheres in their neurons in the substantia nigra that have accumulations of alpha-synuclein. That's the main component of those Lewy bodies. And we've learned from our studies in the pathology of Parkinson's disease that the alpha-synuclein clumps up in an abnormal way, and that's what makes it go into these Lewy bodies. And so we've learned that it's not just the late observation in the disease. It's not just some consequence of a bunch of things at the beginning. It's important at many different steps. We know that Lewy bodies are toxic, and we know that alpha-synuclein by itself, when given to health, when administered to healthy animal models or in cell conditions, can by itself kill cells. So the combined knowledge that alpha-synuclein genetically and then also its role in pathology really implicates a strong role for this alpha-synuclein protein in Parkinson's disease, and that supports our efforts to try to target it. We know that if it's mutated or abnormal, then it's bad. We know that too much is bad, and we know from these pathology studies in the brain where you see these Lewy bodies that there's more of it in Parkinson's disease. So the therapeutic approach then is to try to reduce alpha-synuclein or to try to make it unclump and not be abnormal. So because we think that alpha-synuclein is so important in this regard, the Michael J. Fox Foundation has invested to date over $90 million in trying to understand more about alpha-synuclein biology, understand more about alpha-synuclein biomarkers, and also drug therapies to try to target it. So I mentioned that alpha-synuclein is implicated with genetics and also with pathology in Parkinson's disease. And this is another quick explanation for how we are targeting it as a therapy. Again, I don't want you to be intimidated by the complicated names and titles here. What I want you to understand is that alpha-synuclein normally is in a formation called a monomer. It's just one little sphere, a normal sphere of alpha-synuclein. And then in the disease progression for Parkinson's disease, something happens. We don't know really what that something is, but something happens that causes alpha-synuclein to convert from a monomer to these clumps, to these other versions of the protein that we know negatively affect its role and its normal function, and then they continue to get more and more disorganized and more clumpy and continue to cause more toxicity in the cell until eventually they're found in the neurons, in the Lewy bodies. So we think that there's a therapeutic opportunity here at a couple of different areas. If we can figure out what is causing the alpha-synuclein to start clumping and to start converting from the monomer into the toxic versions, that's one way we could approach it. Also, we could approach ways of maybe being able to convert back from these these different clumps and these aggregates of the protein into the monomer. So, if we're not lucky enough to catch someone here before they, go down the disease process for Parkinson's disease, maybe we can start their therapy further along and help try to improve it back to the monomer. So we think that this combination of the genetics of alpha-synuclein, the pathology that it plays, and now what we know more about the biology of it, the fact that it's a protein that changes over time with the disease, we have a couple of different ways that we can strategize to treat it. So that's what I'll tell you a little bit more about now, is the many different ways that we are pursuing therapeutic advancement targeting alpha-synuclein. So it's, we feel that more shots on goal is really the way to go here. We need many different approaches to try to do this. Whether we try to keep something as the monomer or whether we try to unclump it, we've got a lot of different ways to try to pursue that. To give you an example, this is a quick little biology 101. When you start off with DNA, of course, everybody has the DNA in every cell of their body, and that is what is the template, the program for all of the proteins in your body. So the alpha-synuclein DNA is in all of the cells. It's found everywhere in the body. And this is inside the cell, and the DNA then translates its template to messenger RNA, which really just kind of converts that message of the gene code in your DNA to tell it what protein to make. So whether or not you make a protein for your skin or for your liver, or in this case for alpha-synuclein, that is dictated first by the DNA, then to the messenger RNA, and then it's like a roadmap to finally make the protein. So next what happens is something in Parkinson's disease causes that alpha-synuclein protein to make the toxic Lewy bodies. Once the toxic protein is made through a process that we still don't completely understand, some of it is released from the cell, and then now that toxic protein is outside of the cell, and other cells can take up that toxic protein, and then you start that cascade again of the alpha-synuclein causing more and more cell death and pathology. So one strategy that we have is mopping up that alpha-synuclein that's on the move, taking that alpha-synuclein that's outside the cell and just really kind of mopping it up and getting rid of it. So we actually have a lot of programs and research and drug therapies that we're pursuing with this exact approach to try to mop up that alpha-synuclein. Another thing that you can try to do is to reduce the overall alpha-synuclein. I mentioned earlier that the mutations that cause too much of the normal alpha-synuclein can cause disease, and then over the course of disease, alpha-synuclein accumulates, so too much is bad. So if we reduce the overall levels of alpha-synuclein, that should be a potentially good therapy. So there's a couple of ways to do that. You could either specifically target and maybe, shortcut or, really just cut off that process of converting the DNA to the RNA specifically for alpha-synuclein. Clearly, you don't want to do that for everything else. But if we could figure out the unique process that alpha-synuclein uses, then we could reduce that conversion. And we could also use a similar approach to prevent the protein from being made from the RNA. So there are several different not trials, but early stage research and drug development going on specifically to try to lower alpha-synuclein levels outright. Another potential therapeutic avenue is to break up the Lewy bodies. So if you already have the Lewy bodies present, then you can try to bust those open. But importantly, you would also want to be able to help the cell get rid of those toxic proteins. So if there's a way to it's kind of the mop-up strategy, but not outside of the cell, doing it inside the cell. Your body and your cell has wonderful mechanisms already in place to help, get rid of too many proteins or unused proteins. So if there's a way that we can turn that process on even more in a very specific way, that might be a good therapy as well. So this is I know there's a lot of information on this slide, and I just want to explain what it all comes back to. On the side here is those different ways I just talked about, how we can target the alpha-synuclein, whether by mopping it up, whether by breaking up those Lewy body clumps, increasing the removal of the protein, so that's like the garbage disposal, and then also reducing the total levels of alpha-synuclein, either at the protein or level or the DNA level. At the top here is kind of the pipeline for drug development. So right before the first stage here is actually the very early discovery that happens, and this was the biology that happened, over the last several decades that led us to believe that alpha-synuclein was important. But once a drug company identifies alpha-synuclein as important, it moves on to the stage of lead discovery. That's where they start looking at it and saying, what would be a good way to mop up this alpha-synuclein? What would be a good way to, get rid of the protein? And so there's a lot of research that's going on in the area of lead discovery. Once a drug candidate progresses past lead discovery, it gets to lead optimization, and that's really where they're just fine-tuning, the mechanism of how that therapy might work. Then you move on to preclinical safety and toxicity. So if you've done a lot of work in cell models and in animals at this point, and you've really started figuring out that maybe this is a good drug to move forward, you have to make sure that it has a good safety profile, that you can eventually take it into human tests. And then once you have that information, you move on to those phase 1 and phase 2 clinical trials in humans. And the point I want to make is that the Michael J. Fox Foundation, by putting a lot of this effort into developing drugs for alpha-synuclein, is funding and has a role in many different lead candidates at many different drug companies or even universities and private institutions in each of these areas at all levels of the of the drug development pipeline. The whole goal is to de-risk this middle process. I want to explain that this going from lead discovery all the way into clinical trials and then eventually into the clinic where you can get the medicine from your doctor is a very long and expensive process. Each one of these stages can take anywhere from 1 to 5 years, and it takes millions of dollars. By hedging our bets and placing our bets in all of these different areas, we increase the likelihood of success of something finally moving on and actually getting into the clinic that you can get from your pharmacist. So I want to point out that some of these projects have been directly funded by the Michael J. Fox Foundation. Some of them have been funded by other sources. But there are 2 that are now moving along quite far in this trial process. There's 2 programs that are in clinical trials now in Parkinson's disease patients. And so far, the data is looking good for both of these options. And these are in that area of mopping up alpha-synuclein. So one thing, we've talked about how we need improved drugs that would actually change the disease. I hope I've convinced you that alpha-synuclein is potentially a good target and a good reason to go after alpha-synuclein in terms of drug development. But one of the things that's really holding that back is being able to measure it. So we've talked about how we need to have better outcome measures and a way to track this. So we need to be able to use biomarkers to observe alpha-synuclein in these trials. So I showed you just on that last slide how many drug companies are pursuing alpha-synuclein and how far they are in that process, but we're still working on trying to have a confident way to show that the drug is changing alpha-synuclein levels. So I mentioned earlier that your blood pressure, high blood pressure, would be a risk for heart attack or stroke. And if you take medicine to reduce blood pressure, all you have to do is put the cuff on and see that your numbers are lower. We don't have something like that for alpha-synuclein. It's a very tricky protein. Sometimes it's inside the cell, sometimes it's outside the cell. We really need to develop ways to be able to measure it to allow the drug companies and the clinical trials to have a definite answer. Did it work? Do you need to treat longer? Do you need a higher dose? We need to improve the biomarker for alpha-synuclein so that we can actually answer those important questions. So if we figure out a better way to measure alpha-synuclein levels, this may help us track the disease better and also inform how to design those clinical trials. I mentioned, it might need a longer treatment or a different dose. One of the ways, Dr. Vaillancourt's already showed you how important brain imaging is for looking at changes in the brain for Parkinson's disease. If we could find a tracer that was specific for alpha-synuclein, it only binds alpha-synuclein, then if you take the drug that's supposed to lower your alpha-synuclein, you should be able to see that signature, that brain imaging change. But sometimes it's hard, it's hard to actually get a tracer that's really specific for just one thing in the brain. So we're also working on other ways of measuring alpha-synuclein. We're checking whether or not you can measure alpha-synuclein in the spinal fluid, because I mentioned it's found in other parts of the body too. But importantly, We have found a way to measure alpha-synuclein in the spinal fluid, but we need to be able to confidently correlate those levels that are in the spinal fluid to what's going on in the brain. Because if it's not something that changes, either with the disease progression or in response to a drug, then it's not really going to help. We're also working on looking at alpha-synuclein biomarkers in all of these other areas. So alpha-synuclein is found in the colon and in the GI tract. You can actually measure it in the skin. You can do a biopsy and measure it from salivary glands. You can also get cell lines that you can make from the skin. You can convert that into cell lines and measure alpha-synuclein that way. You can also measure alpha-synuclein in the blood and plasma, and we even have some projects that we're funding using these specialized instruments to measure alpha-synuclein in the retina. So the idea here is that eventually maybe a signature of alpha-synuclein can be identified. It would be great if it'd be as easy as a blood test. Maybe you don't have to get a spinal tap. We feel that we really need to look in all of these areas in a very holistic, rigorous way to find a biomarker for alpha-synuclein that can be used for these trials to test whether or not it's changing the alpha-synuclein and hopefully modifying the disease itself. So this is actually some data looking at alpha-synuclein in spinal fluid. This was data that came from that Parkinson's Progression Markers Initiative that Dr. Vaillancourt was talking about. This is the PPMI for short. This is the flagship biomarker program that the Michael J. Fox Foundation, in cooperation and partnership with more than a dozen industry partners, it's the largest Parkinson's biomarker study on the planet. It has hundreds of patients across the globe, hundreds of clinical centers, and it's what we call a longitudinal study. It's where a patient will first go into the study and they have a bunch of tests that are run at the beginning. They take various biofluids that includes those ones I mentioned on that last slide, that includes blood and brain imaging and urine and spinal fluid, all of these different biofluids. And then other people can study different things in those different fluids and tests to look for changes. So in this case, we wanted to see how alpha-synuclein levels compare in Parkinson's disease patients, that's in the orange bar here, and in healthy control, that's in the black bar, in the spinal fluid. So if you look at the value at the beginning when the study started at enrollment, you can see already at enrollment there was a significant difference between the alpha-synuclein levels in the Parkinson's disease patient and the healthy control. We were a little surprised actually that the alpha-synuclein levels were lower. We don't know what that means, but we do know that there's this distinct difference. What's really interesting is that when we looked later at 6 months and 12 months after enrollment, those levels didn't change. There's no difference between the healthy control over time. It stays the same. The Parkinson's disease patient also stays the same, but there's that distinct separation between the two groups. The big question here is if you were to test these patients after they had taken a drug that's supposed to target alpha-synuclein, say that drug to mop up the alpha-synuclein, would these levels change? Would the Parkinson's disease patient look more similar to the healthy control if you look at alpha-synuclein in their spinal fluid? This is what we're working on all these studies and how we want to come up with that biomarker signature so that we could actually have some confidence when we get further along in these clinical trials. So I want to wrap this up by really, I think that this is a really tricky question, right? I mean, it's not because smart people haven't been trying for decades to find drugs that are a better therapy for Parkinson's disease. It's that it's a really challenging process. I showed you that we have a lot of efforts ongoing and we're supporting a lot of research in the millions of dollars to, try to develop a disease-modifying drug and also to develop better biomarkers. And I want to kind of bring together the progress that I think we've made. We're moving forward towards a therapy that slows or stops Parkinson's. This is through a combined approach of looking and improving various aspects of this entire process, looking at improving tools and biomarkers so that scientists and doctors have better resources and more consistent access to the things that they need to be able to measure changes meaningfully. That includes therapeutic development, working with industry partners, helping to de-risk that middle stage of drug development where many drugs fail to move forward. and also by understanding more about the biology and enrolling patients into clinical cohorts. That's what we call patients that are specifically identified to be good candidates for a trial, getting those cohorts of patients together so that that this entire infrastructure can be built for improving the likelihood of success of drug trials. So some recent progress that we've made in the tools and biomarkers area is that we have been working really hard on generating and sharing these research tools. Dr. Valencourt mentioned that any doctor or many people can access and research some of the data sets that are in PPMI. If we were to develop an alpha-synuclein imaging tracer through the efforts of the Michael J. Fox Foundation, we would make sure that it was highly accessible. We would make sure that anybody could really get a hold of it. It wouldn't be proprietary and held within one company. It would be widely accessible. That's why we're leading those efforts to visualize alpha-synuclein in the brain. And we're also working on developing the protocols and tests necessary to measure the alpha-synuclein in the biofluids. Some other highlights and progress that have been made in the area of understanding the biology and the clinical cohorts is in recruiting an alpha-synuclein genetic arm to our Parkinson's Progression Marker Initiative. That's that big PPMI biomarker group that originally just had patients who met certain criteria, and we recognized recently that with this new effort going on in the alpha-synuclein area, that identifying patients who have the mutations in alpha-synuclein might be a good way to make sure we can study that group as well. We're also learning a lot more about the normal role of alpha-synuclein. I mentioned that if it's mutation, it causes the protein to behave incorrectly, but also just having too much of it. So we're studying a lot about that process, and we're developing models that we hope better mimic the human pathology so that we can test things earlier. In the therapeutic development area, I want to highlight that there are two drugs right now in clinical testing that are pretty far along in this trial process, and 3 more that will potentially be in later stage clinical trials in early 2016. We're funding more than 25 different drug discovery programs on alpha-synuclein alone, and hopefully, the whole idea is by putting more shots on goal, something will come out sooner. So the last thing I want to talk about is how you can get involved in some of these trials, and I hope I've emphasized how important some of these studies are for biomarker development and for getting improved drug therapies. And you have a great resource here at Pennington with the trials that they have as well. So I want to just quickly mention Fox Trial Finder. It's an online tool that the Michael J. Fox Foundation has where trials and sites all across the world can post their trials on the website, and it uses a matching process. What you do is you go on and you enter your information and set up a profile on Fox Trial Finder, and then once the online tool then matches your profile with the criteria of the different trials that are ongoing. And what's great about this is it also can match this by your geographical area. So for those of you who traveled away, or who aren't from Baton Rouge, or who traveled from further parts away, or if you move to another part of the country, if you update your profile, then the Fox Trial Finder tool will help you find trials in your area. The other thing that it can help is sometimes there are trials that can be done with like video conferencing or through other platforms that don't require travel or local proximity to a clinic site, and so this can help with that as well. So currently there are over 45,000 volunteers enrolled on, have profiles on Fox Trial Finder, and there are more than 400 actively recruiting clinical trials across the globe. So if you want more information, go to the Fox Trial Finder and certainly look into the information that's available locally here through Pennington as well. The last thing I want to mention is Fox Insight. This is a new platform that the Michael J. Fox Foundation has recently embarked upon. Fox Insight is a study. I want to emphasize the difference between a trial and a study. A trial is where an intervention or a test is being evaluated. So whether or not it's exercise or diet or a drug, a trial is looking at whether or not that thing being tested is changing something. A study is more like what that PPMI study was, where you're not putting in a drug to change something, but you're really trying to get information about what's changing over time and how those biomarkers may be shifting. So Fox Insight is a digital tool for us to try to get more information through a study. It uses things like the Apple Watch or your cell phone to be able to record movement. So So the same way that your cell phone can tell you by GPS where you're going, your cell phone might be able to tell whether or not you had a fall or whether or not your tremors are worse on a certain day. It can measure whether or not it's the medicine is kicked in. It's a good way for us to be able to collect information directly from patients, and it goes into a data storing system that can then be shared with doctors. It goes into an online system that can then be accessed by researchers to look at other information as well. So there's more information on Fox Insight also at the Michael J. Fox website. And I want to emphasize that Fox Insight, because it's a study, anyone can participate in it. Everyone in this room is eligible for Fox Insight because it takes healthy controls and anyone who has Parkinson's disease. So I welcome you to take a look at that. Lastly, I just want to give some acknowledgement to my colleagues at the Michael J. Fox Foundation for all of the hard work and the efforts that we have. It's really a very dedicated and motivated team. And our alpha-synuclein portfolio area has a lot of momentum and a lot of inertia and a lot of really, really bright people working on it. It really does take a village, as they say. So specifically, the people circled in green are the ones who directly contribute to our alpha-synuclein portfolio, and that includes Mark Frazier, my colleague Koldup, Jamie Eberling, who works in so Koldup leads our alpha-synuclein therapeutic initiatives, Jamie leads our alpha-synuclein imaging initiatives, Samantha Hutton leads our alpha-synuclein biomarker initiatives. I support all of these groups by improving the tools and industry engagement with the industry groups. Allison Morris is our officer and our business strategist who helps with all of our alpha-synuclein programs. And also want to mention that we really do have a team approach at the Michael J. Fox Foundation. Every priority area that we identify has scientists and business strategists working as hard as possible to move forward as quickly as possible because we need better therapies today. Thank you.